What's that?
Metamerism involve organs and specialized.
Well, they both involve organs. So what's the difference between segmented organism and organisms that are segmented and metameric?
There's a specialization of organs within each section.
Or a section could become specialized for specific organs. So in basic segmentation, the idea is that each segment is a repetition of the body part, right? And they might have all of the same parts in that segmentation. Metamericism is a modification of that segmentation in such a way that individual repetition parts end up being focused or specialized for certain things. So if you notice like this annelid, right? The annelid has each one of these rings represents a segment. And each one of these segments, when you look inside down the trunk of the body, has pretty much the exact same stuff. A pair of gonads, a pair of nephridia, a coelomic space. The gut runs through them all, but pretty much each segment is the same thing, just a repetition of the same thing. In an arthropod, those segments are clustered together for specific purposes. So you have a group of segments that become the head. Like in insects, there are 5 segments fused to form the head. And each one of those segments has an appendage, but those appendages, the first 5 appendages, cluster together to form the mouth parts. That is metamericism. They're grouping in such a way that a cluster of segments can form a functional organ. So it might be within the body, an organ in the body, or it might be a group of segments together forming a swimming appendage. Segmentation. So, our understanding of the evolution of animals has been changing and is a bit more complicated than we used to think. The story that I learned as an undergraduate 25 years ago was very simple. We went from having acoelomate, first we went from having diploblastic organisms to triploblastic organisms, so the move from, we'll talk about the groups. We talked about the development of acoelomate to pseudocoelomate to coelomate interior cavities, that was a progressive sort of evolutionary change. We talked about things like segmentation arising multiple times in different groups. The traditional way that we looked at this organization was all based on morphology, right? The progressive move of no coelom to a pseudocoelom to a true coelom. And the phylogenies that we built, phylogenies being a graphical representation of the evolutionary relationships of these groups, put together this very clear sort of progressive story. And it turns out to be the case, once we start looking at molecular data, that's not so true. So if we look at sort of the traditional way that we did this, we would look at a series of character states. These are attributes about organisms, and then we would look to see whether or not they have them or they don't have them. And that's the cool part about building phylogenies from morphology. You say, does this thing have this character state? And you build as many things as you possibly can. You build in as many character states as you possibly can to form your tree. So in this tree, we have 3 taxa and 3 character states, and that's it. You can add in as many character states as you can conceivably think up of, and you can add in as many taxa as you have character states to know for. And basically, you just use this matrix of character states. Usually we put it in a binary setup. You either have it or you don't have it, so it's either a 1 or a 0. And from that, you build a tree that is able to account for all the data that you have. Sometimes you can get competing trees, and there are ways to statistically determine which one of the competing trees is more likely. And you get a tree, right? So in this case, you'll notice that at each one of those little greenish circles, that is when that trait arose evolutionarily and is shared by everybody in the tree after that trait arose. So if we're going to look at 2 paired appendages, whoever was the precursor for sharks didn't have 2 pair appendages. So if you look at something like a lancet, which is a primitive chordate. It doesn't have appendages at all. So two paired appendages showed up with sharks, and then every organism afterwards along the phylogeny has it. Digits, fingers, showed up at a point that every organism on the phylogeny has it after. And hair showed up only once on this phylogeny with the mammals or humans in this case. So that's how you construct trees. Phylogenies are hypotheses of those relationships. They are not the actual relationship. If you think about it, none of us can actually be there when it happened to really know exactly what that relationship is. We can infer those relationships from the data that we can take. So phylogenies are inferences that offer us a hypothesis that help explain what those relationships between the organismal groups are. So what's cool is that this works just as well with molecular data, right? So rather than having a character state of does it have it or does it not have it, with molecular data, we end up with 4 character states, each of the potential nucleotides that it could have in that spot. And if you look across a data set of molecular data, every single aligned location in all of those sequences act just like one of these little character states. If they all share the same letter, that means they shared a common ancestor that had that same letter. So, traditionally we've split animals into 2 big groups, right? Parazoa don't have symmetry and they lack tissues, and the eumetazoa, the true animals, have symmetry and tissues. Biggest fundamental split between everything in the animals. The next split between those that are eumetazoans are those that have a 2-germ layer development and those that have a 3-germ layer, or a diploblastic and triploblastic. The total group of metazoans are split out into around 35 phyla, though that's changing. And for the most part, we have a phyla that is parazoa, a phyla that is no tissues, no symmetry. One phyla that is diploblastic, and all the other 33 are in the triploblastic group. Okay, so this is how I learned the classification when I was a youth. Okay, it looks reasonable enough, right? We have acoelomates. All right, so we have our no tissues parazoa at the base. We have our diploblastic groups. Turns out to be not the case. One of these is actually triploblastic. We have diploblastic groups. We have an acoelomate group, and then a pseudocoelomate group, and then everybody else on the tree, this big group here, based on this node, this point on the tree that's shared, are all coelomates. There are coelomates that have development that is determinate, and that is indeterminate. So we have our 2 big splits of deuterostomes and protostomes. Made perfect sense in the data that we were looking at. This is what I learned as the traditional classification. In fact, the textbook that I had sort of worked through each of these groups in this way, showing here was the arising of a pseudocoel, here was the arising of a coelom. Yeah, it turns out to be when we've added in the molecular data, that's not quite the case. And there have been some changes that took place once we've looked at what that molecular relationship is. And this is the new way to see it. Notice that the gut space, or the coelomic space, has become less important in determining what those relationships are. The movement of acoelomate to coelomate to pseudocoelomate to coelomate has sort of disappeared as being important. And what was really remarkable is once we started looking at the data that was molecular, we saw relationships that no one had thought really were that strong. So for example, notice who is sister, that's the next back branch to the arthropods. What is it? The nematodes. When I was a undergrad, annelids and insects were grouped together. annelids and the arthropods, both segmented, both protostomes, both true coelomates. Nematodes are pseudocoelomates. And only after the fact that we recognize their relationships molecularly were we able to figure out some unifying thing that brought those two together in a fundamental way. Both of those groups, the nematodes and the arthropods, are what we call the ecdysozoans. They shed their skin. They lose their exoskeleton, or in the case of the nematodes, a cuticle, under the direction of one kind of hormone called ecdysone. So after we figured out molecularly that these guys were related, we figured out a fundamental trait for that group that is shared by no other group. The ability to molt or to shed your skin under the control of a particular hormone. The other thing that was really amazing is that mollusks and annelids had always been thought to be not closely related, which is kind of funny when you think about their larvae, because their larvae look exactly the same. Their larvae are called trophophore larvae. They look identical. There's really only one set of 4 cells that if you look at in their larvae, you can tell that one is a mollusk and one is an annelid. So after the fact, now that the molecular data groups them together, you go, oh, hey, think about it. One of the most fundamental traits of this thing, the development of its larvae, are very similar between the two. And so now we've made that group actually cluster together based on the larvae. Trocho, you see that name in there, right? The trophophore is the name of their larvae. But they also have this little feeding tufted appendage on the top of their larvae that look a lot like the feeding appendage that's called a lophophore of this whole other set of groups. And the realization was that the lophophore and trophophore larvae actually represent a shared characteristic across this whole group. So they're now called the Lophotrochozoa, the animals that have trophophore larvae or lophophores. So those changes are pretty big in our understanding of the evolution of animals, because it looks like things like the development of a pseudocoel may not be a direct line to the development of a coelom. In groups like the annelids and the other Lophotrochozoa, it looks as though the pseudocoelom state might be a reversion to a previous kind of thing, or it might be a side branch. So some of the basic outlines are still the same. Sponges separated early, right? Sponges represent a very early animal group. The diploblastic, radially symmetrical represent the next sort of major split of animals. And then the bilaterians are split from that. So we still have the same basic three big groups, right? The no symmetry, no tissue parazoa, the diploblastic group in the metazoa, and the triploblastic group in the metazoa. But the relationships of some of those phyla within the metazoa, within the bilaterians, is a little bit different. So the biggest difference really is that nematodes and arthropods are now grouped as a single group, the ecdysozoa. Okay, so now that we've got this whole set of data, molecular data, physical characteristic data, the new modern phylogeny looks something like this. Rather than having a move from coelomic spaces being what really is the biggest idea in grouping these things, the thing that groups them the most individually are often specific traits about specific groups. So check out the ecdysozoa. The nematoda has a cluster of related phyla. We're not going to talk about most of them. The Loricifera are really, really cool, but we're not going to talk about them. What do you think that whole group shares? The ecdysozoa. We've already talked about the arthropoda, and we talked about the nematoda. Look at they're like on 2 separate branches. What do you think every single organism in that group shares? Molting under the control of ecdysone. That's the common trait of that entire group. No matter what their bodies look like, they all have ecdysone that is part of their molting process, that controls the loss of their exoskeleton or cuticle. Okay? What about that Lophotrochozoa group? Notice that they're all clustered together. Lots of those groups used to be thought of as being highly divergent. In fact, at one point, if you look at the Bryozoa, the Ectoprocs, and the Brachiopods, that group used to be thought of as being a transition to the Deuterostomes and had some Deuterostome-like traits. Now it's clearly in the Lophotrochozoa. The molecular data put it there. What is their common thing? Not all of them are segmented. The larvae and the lophophore, the feeding appendage of the larvae and in some cases the adults. You'll see there's also now a group called the Platyzoa. We're not going to talk about every single one of those groups in there. The idea that Platyhelminthes, which are the flatworms, the acoelomate flatworms, represented a bridge or transition to the group that was pseudocoelomate, doesn't appear to be the case. We now end up having some groups very clearly together, the rotifers and Gnanthostomidia. One of those is a pseudocoelomate, one of them is a true coelomate. The Platyhelminthes is an acoelomate, but the Cycliophora is a true coelomate. So the idea that coelomic spaces are really what drove the evolution of animals doesn't appear to be so. Now, why did we tell you about them? Why did I tell you about coelomic spaces if it's not what drove or was not a big thing in the evolution of organisms? It still helps us understand how organisms are able to do what they do. It helps us understand how organisms evolved particular kinds of responses to difficult things in the environment. And it helps you understand their body plans, what we call Bauplans, is a German word that's sort of more than just what their body plans is. It's like the entire gestalt of the organism, the way that they do everything they do is all wrapped up in that word. It helps us understand the peculiarities of each group's little issues. If you're an acoelomate, there's only certain things you can do. For example, you cannot sequester nitrogenous waste in a coelomic space because you don't have one. You have to come up with some other way to get rid of the nitrogenous waste. So, one of the reasons why we tell you this story is because it opens the door for you guys to see how science actually works. 25 years ago when I learned this, it was textbook, literally. And it turned out to not be so. One of the things that science is about is leaving the door open to new data that helps you change your interpretation of how things work. 25 years ago, if you told me 25 years from now you're going to be teaching college and you're going to be teaching something different than this, I would have told you to go jump in the lake. But the fact is, as a scientist, you leave it open for new data to come in and change your view. And when the data came in and you look at it and you go, oh yeah, okay, so every single gene that we look at puts annelids and mollusks together and doesn't put annelids and arthropods together, that wouldn't make any sense if we stay with the old phylogeny, and so we better change the phylogeny. This is currently happening still with invertebrate groups, and it's currently happening still with, especially, protist groups. But right now, there's actually a worldwide global effort called the ATOL project, which that's not atoll out in the middle of the ocean. That is the analysis of the tree of life, right? Where we're trying to decipher the relationships of every single group of animals that we can, and all groups of organisms. And the relationship of them and the clarity of where their relationships are is coming into much better focus now that we're able to combine molecular with physical attributes. So, now begins what we like to affectionately call tiptoe through the phyla. You have been given this overarching cap of the way to view these organisms. These organisms do not sit in isolation as like, oh, well, this is this group, and this is this group, and this is this group, right? You want to think about them going back to those 5 big themes. Where do they fit within? Symmetry, Where do they fit with the kinds of germ layers they have? What about development? What about, so all those things that we talked about, okay? That you want to think about each one of these groups in terms of those big themes. You want to think about these groups in terms of the phylogenetic tree. Use the modern one to understand where we think they are now, but recognize what about them suggested their relationships in the traditional classification. So, our first group is going to be the Porifera. Porifera means the bearer of pores, the pore bearers. That is the sponges. Sponges are kind of like animals. We call them the parazoa, or the near animals. They have some animal-like traits. They have, so their fundamental animal-like trait is they are heterotrophs that get their energy by ingesting or engulfing the organism or the things that they're eating. Okay, so how do they do that? They don't have tissues. They don't have musculature. Actually, they do have some cells that are called myocytes that actually act like muscles, which is kind of cool. They don't have a brain. They don't have nerves. How do they ingest something? Well, this group are essentially a loosely organized group of cells living within a non-living, living on and around a non-living matrix. So what they do is sort of secrete a structure upon which individual cells live. And those individual cells are colonial with the other cells. That means each individual cell exists independently of the others. There's not a unified structural set of cells that form tissues. Each cell is kind of on its own. But they work together with each other to be able to keep the organism fed and alive. So what do I mean by each individual cell is on their own? If I take a sponge and put it up against a mesh that's one cell big and squish the thing through it so that every single cell passes through the mesh having been disassociated from any cell that's nearby, push it through the mesh and get to the other side, the pile of cells on the other side would spontaneously reform into a new sponge. There's some really cool experiments that have done this. Each cell recognizes the neighboring cells around them as being part of their unit, but each cell is independent of all those other cells and can actually exist on its own, but will form a new sponge when it's in contact with a bunch of other cells of its own origin. Pretty awesome, huh? Right? So, can you think of any other animal that you could squish through a sieve and on the other side they'd all regenerate back or form back into a full-grown organism? No, because there aren't any others that do that. So, all of these organisms are sessile, which means they live attached to the bottom. And there's only about 5,000 species or so recognized, although we are applying molecular tools to them, and things that we had thought were one species are turning out to be cryptic species, where you go from, 3 meters and down to 20 meters, this organism lives, and we'd always thought it was one species, and when you start looking at it, you realize that it's partitioned out in different species each 5 meters or so you go down. So the number is probably more like double to 3 times as many sponge species as have been named, probably maybe even more. Almost all of them are marine. Almost all of them live in the ocean, although there are a few that live in fresh water. So, the basic setup of a sponge is that of a vase that is one cell layer thick on the inside of a non-living matrix and one cell layer thick on the outside of that matrix. This body plan exists like this, usually in really small size, and we'll explain why that happens in a bit. So the basic setup is running on the inside of the vase shape is a set of cells that are called choanocytes. what was the name of the group that we think our protist, most closely related protist is? The Choanoflagellata. In fact, the choanocytes on the inside of the sponge look exactly like the Choanoflagellata. Those exist independently as one cell on their own, but you look at a sponge and the inside is covered with these collar cells that look just like choanocytes, or look just like the choanoflagellates that are free-living protists. But these exist as a colonial set. So that's one clue that suggests that sponges are related to choanocytes, and the Choanoflagellata are our colonial ancestors. So, running on the inside here, you've got this set of cells that are all got a little collar on them. They've got a beating flagella. When all those flagella beat together, they are able to form a current that moves up along the side of the cells. And so water flows up and out this top part, which is called the osculum. And that makes water get pulled into the inside of the sponge through holes that are along the side of the sponge. Those holes are called ostia. The flow of water coming through, formed by the choanocytes, helps to draw food particles into the interior of the sponge. They're living in this marine system where there's just all kinds of suspended things, algae, particulates, dead and dying material that they can feed upon. So this interior is lined with choanocytes. Those choanocytes are not actually connected to each other. There's actually little spaces between them. Notice that there's no like basal lamina here. There's no place where the cells, remember when we talked back in 200 about things like hemidesmosomes, those like basal lamina sitting along the bottom of a set of cells that they're all stuck to, that if anything's going to get to the other side of that, they have to actually go into a cell. Tight junctions between two sets of cuboidal epithelial cells make it so that nothing can get from one side of the epithelium to the other without going through a cell. This doesn't have that. This is just cells butted up next to each other. And it's actually what we call an epithelioid. Fluid can flow between these two cells without ever actually having to go through a cell. So there is actually no inside or outside to this organism. Each cell is kind of on its own. So as we work our way through the wall, you've got this inner layer on the inside called choanoderm, made of choanocytes. You've got this non-cellular matrix between the two, which is basically like it's called mesohyl. It's often got things like proteins that have been secreted, stuff that's called like spongin. There are cells in there that can form spicules, little calcareous or glass spikes that form the skeleton combined with the spongin, combined with the protein. And you have an occasional cell working around inside that matrix, but for the most part it's non-living. And then on the outside you have this layer of flat cells that are called pinacocytes that form a pinacoderm, an epithelioid. So the structure of this thing is incredibly simple, just a couple of cell types. In the middle in the matrix, there are some cell types that are called archaeocytes that can form any of the other cells. So if a choanocyte dies, an archaeocyte will come down there and form and replace that cell with a different cell. All right? So notice that they're sort of tissue-like, but not actually tissues because they're not combined with each other. The way that they feed is actually really cool because there's only one cell that actually digests food. The choanocytes flop their flagella, they form a current on the collars. If they catch a food particle, they pull it in and hand it off to another cell. They actually take a little vacuole and make a vesicle around it. They carry it through the choanocyte and hand it off to an archaeocyte that does the digestion. So it's only those archaeocyte amoeba-like cells within the mesohyl that do the digesting. Individual cells don't actually digest for themselves, which is kind of bizarre. If you come across a particle that's coming through the sponge that's not edible, it gets entrapped by a pinacoderm or entrapped by a porocyte, which is the cell that lines the ostia, and it hands that particle off to an archaeocyte, which then carries it through the mesohele and dumps it off on the other side. So non-living particles that aren't edible also get taken care of by the archaeocytes moving it to the other side. And things that are edible end up getting brought into archaeocyte that then does the digestion. So it's a filter feeder. It's basically pulling out of the water column suspended particles that are edible and then handing them off to something that can digest them. Kind of cool, huh? So what ends up happening is that this organism is not static. The shape of it is not static. Pinacocytes die after a while because they can't get food. It's only the archaeocytes that are getting food. Choanocytes along the inside die after a while because they can't get food. And so the archaeocytes are actually constantly replacing the other cells. The archaeocytes are like the stem cells, but they're always replacing the other cells as they end up dying. That also means that the organism is incredibly dynamic. You wouldn't think a sponge is actually dynamic, but they can change their shape. They can grow new places and new parts because when some cells die, new cells can come in and replace it. When we see a little more complicated body plans, they start getting pretty bizarre in the things that they can do. Now, they are sexual organisms. They make sperm and egg, and they throw that sperm and egg up into the water column where they form a zygote. So they are on their way to becoming animals, really. They've got the haploid sperm and haploid egg that fuse together to make a zygote, and that zygote, for a cell, starts dividing. But those cells don't form tissues. They just sort of stay around near those early cells and form a big cluster of colonial cells. Many sponges can reproduce asexually as well. They do it in a couple of different ways. One, like a wave comes by and rips off a part of your sponge and it lands somewhere different. That thing can form a whole new sponge, will grow up into a normal sponge shape. But under really stressful conditions, sponges can make these little asexual balls of cells. They sort of like cluster a bunch of cells together, make some spicules, those are like the calcareous or glass spikes, into a sphere around those things, and then they set that thing off. These are called gemmules. The gemmules sort of fall onto the surface of wherever they're a new location, and then it'll crack open, and all of the cells come pouring out of this micropyle and form a new cluster of cells that's going to form a new sponge. So they can reproduce asexually by fragmentation. There are some things where sponges will just tear themselves in half to form two new sponges, which is kind of cool. I've seen it in slow-mo where someone's filming the sponge for a long period of time. You just see it's like pulls apart and then reforms back into two sponges right next to each other. It's pretty cool stuff. Okay, so they have both sexual and asexual reproduction. Most of you guys have seen sponges that are large, right? The sponges that you think of are these things that are, I don't mean like the sponges you use in your sink, although there are sponges that are actually called commercial sponges, which is what the sponges in your sink are based upon. They're actually based upon a real sponge. What happens is that there is a size limitation to this body plan. The size of that little basic, vase shape cannot get too big because only a certain amount of water can actually be moved by the choanocytes. Choanocytes can only move water about, half a millimeter to a millimeter away from the surface of the choanocytes. So the most that you see for the basic kind of shape, which is called an ascon, is usually only about a millimeter across and about a centimeter high. So you end up having a limit to the size of what you can get for your ascon shape. All right? So how is it that sponges can get bigger? if there's this fundamental limitation to being able to move fluid through the body plan, how is it that they get any bigger than this little 1 centimeter by 1 millimeter sized sponge? If you look at, if it just got bigger and bigger and bigger, keeping that single layer of choanocytes on the inside and a single layer of pinacocytes on the outside, there wouldn't be any water movement in the middle of the inside of the sponge. This part's called the atrium. And there wouldn't be enough to flow water out the top of the sponge. There wouldn't be enough movement to pull water in that can feed enough of those cells. The fundamental limitation here in terms of the size of an individual chamber is this little 1 millimeter to 1 centimeter long thing. So the way that the sponge deals with that, the way that it's evolved so that sponges can get bigger, is to basically just stack the ascons one on top of each other. So you could imagine it like, if this is your ascon, giving you one little shape here, if you just stacked your ascons like this, around in a shape like so, I'm going to draw in little choanocytes on the inside here so that you can see that there's flagella on the inside of these things, okay? And then made it so that water could flow in and then into the individual into the individual chambers. Each one of these still is the 1 millimeter wide, 1 centimeter long shape. But they've now been stacked onto each other, and if you sort of loop back between these things and form a series of chambers, all that are lined in choanocytes, you basically kept the same physical limits to how big it can get in each individual choanocyte chamber. And this setup is called a synconoid or a syncon. Notice that it's going back and forth in this sort of sinuous way with only flagella on the inside of each one of these chambers. Okay? Most sponges that you think of, big sponges, are actually lots of these things, lots of chambers. Each chamber is still within are still within this size range, 1 millimeter or so, but they're in these large bodies where there's this whole set of canals that run through to meet an individual choanocyte chamber, and that choanocyte chamber is lined with choanocytes. And it's all over the place in there. In fact, leuconoid sponges are really dynamic because those choanocyte chambers, remember, those choanocytes are dying when because they're not feeding themselves, right? Those things have chambers shift and move all the time. New channels come through. This whole entire system is called the aquaferous system. And so the interior of a sponge is always rebuilding itself and breaking down old portions and making new canals and new shapes and new chambers interiorly. They don't look like they're dynamic, but they're actually incredibly dynamic over time. So these are our 3 basic shapes. Synconoid are the middle size based on a series of ascons. Leuconoid have choanocyte chambers that are still within the size range of a asconoid shape, but just all over the place inside of the system. And all of those choanocytes beating at once can move a lot of water through a synconoid sponge and through a leuconoid sponge. They can bring a sponge that's 1 liter in size, a leuconoid sponge that's 1 liter in size, can move a liter of water through it in just a couple of minutes. So a sponge that's this size can move hundreds of gallons a day through its system, picking out individual particles that are edible and moving them to the cells, to the amoeba-like cells, amoebocytes within the mesohele that are able to digest it. So today in lab, you're going to look at some of these things under a microscope. You're going to be able to see the choanocyte chambers and see sort of what these cells look like. They're really, really small. Okay, so there are 3 major groups of sponge. Mostly what you think of when you think of sponge are demo sponges. Those are the ones in the middle. There are a group of very small freshwater sponges that make their spicules exclusively out of calcium carbonate. Those are called calcareous, the calcareous sponges. And there is a group that makes their spicules exclusively out of glass. And they actually do it in this really cool way because they don't have cells per se. They form a structure, a matrix of glass, and then surrounding that glass matrix is one single cell that spreads across the entire surface of the glass. There is lots of nuclei in it. Each nuclei controls the expressions of proteins right near the nuclei. So it forms basically a giant multinucleate cell. Can you see why we thought that the multinucleate hypothesis for the formation of animals might be that way? Because there is this group. That group are called the hexanactinellida. Say that 5 times fast. We're going to look at one of those glass structures in lab, and they've got these really cool-looking five-sided structures that the glass all lines up with. It's actually glass. But it's a syncytium. That's what we call a multinucleate, single, unified tissue-like structure, a syncytium. And hexactinellida is a syncytium. One single connected cytoplasm, one single membrane across the entire thing, forming a multinucleate giant cell attached or surrounding a glass matrix. They're awesome. We'll look at them in lab. Boy, this is tough. When I teach invertebrate zoology, I spend like 15 minutes talking about the hexactinellids because they're cool. All right, so that's the sponges. The sponges are our first phylum. Parazoa, simple structures, no true tissues. They're not even in the diploblastic, triploblastic thing because they don't have two tissues. The next phylum we're going to talk about is really cool because it's got one species known. That species was discovered in the bottom of a fish tank in the 1880s. And it looks like an amoeba. It looks like a single large cell crawling around. But when you look at it close, you realize it's multicellular, first of all. And what is really cool about it is that it forms this creeping soul, forms this creeping set of cells. Those cells are actually connected together with tight junctions, which is kind of a new and interesting thing. But it's got 3 cell types. It's got a single cell type that's growing on the creeping soul, a second cell type that's growing on the top, and these interior middle cells that are a syncytium. So it's almost halfway between colonial and syncytial. The way this thing lives is it creeps along the bottom of sediments. And when it finds an edible particle, it crawls up onto the edible particle and then wraps around it, forming essentially like a little string purse. So it's a flat thing that curls around and it forms a little opening that then closes around this food particle and then floods the interior cavity with digestive enzymes. Digests the thing inside of there, moves it across, moves those digestible fluids off onto the cells that are that are lining the inside of that bag, and then once it's done digesting, it lays back down flat and creeps around to find another source of food. So why that is interesting is that we've gone from having no tissues to having 2 tissues, and the fact that it wraps around the food particle forms an interior surface that is where digestion is located, and an exterior surface that's essentially the protective outside part, which is exactly what we see in the next in the next phylum. So a epidermis on the outside and a gastrodermis on the inside that can be used to digest. That is what we see in the next group, which are the cnidarians. This has got a silent C, cnidarians. The cnidarians are the jellies, sea anemones, corals, and there's a few other groups that we're not going to talk about. They're really cool, including some deadly ones. The little, this guy, this is a cubozoa, this guy is deadly. These things kill more people than sharks every year, by an order of magnitude. How many people were killed by sharks last year? 18 last year, yeah, in 2016. 18 people globally killed by sharks. How many sharks were killed by people? A lot. Like twice as many? 10 times as many? 10,000 times as many? About 100,000 times as many. About 200 million sharks were killed by people last year. And 18 people were killed by sharks. Who is the vicious predator on this planet? I'll tell you who it is. The cubozoa. Those guys killed about 250 people last year. Almost all of them in Australia. All right. This guy is a stalk jellyfish. They're really cool. All right. So they are eumetazoans. They have true tissue. Their individual cells within their tissues are unified together with tight junctions, with a basal lamina. In order to get from one side of that set of cells to the other, the only way to do it, if you're a particle, is to go through a cell. There's no spaces between the cells to push from one side to the other. That means that it is a true epithelium. There is now really an inside and outside of this organism. So, it has distinct tissues, but it doesn't have true organs. So what do we mean by true organs? What was the definition of organ when we talked about the hierarchy of cells? Tissues unified together to form a single structure used for one thing. And we don't actually see that in the Darians. There aren't things like kidneys. There is an outside layer of cells and an inside layer of cells. And if you were to say form a gonad, it forms from one of those inside or outside layers of cells. You never get a multicellular organ structure inside these organisms. All there is that's living is the outside layer and the inside layer, or the epidermis and the gastrodermis. So, what's between the two of them, like we saw in the sponge, is a non-living acellular matrix. So like a connective tissue kind of thing. In sponges, we call that layer the mesoglia. Meso means middle, right? So in sponges, it's a mesohele. In cnidarians, it's a mesoglia. And that is the jelly of the jellyfish. Now, it's not like, Jell-O or something. The jelly can actually be pretty rigid and stiff. But it's a non-living acellular matrix. There might be an occasional amoeba site. There might be an occasional cell sort of moving around within that non-cellular matrix. But the jelly that makes up the jelly of a jellyfish, we don't like to use the word jellyfish because they're not fish. They're just jellies. That is non-living. So, these guys have no brain. There's no centralized nervous system, but they do have nerves. Those nerves exist as a nerve net, one running around the outside in the epidermis and one running around the inside in the gastrodermis. So they can coordinate movements across this nerve net, but they don't coordinate them in a way with the brain, okay? So that means no cephalization. Their gut is incomplete. What that means is there is no mouth end and anus end. There's just one opening. There is a single opening. So you can think about their body as essentially just a bag, right? You open up the end, you put food in, you close it. Anything that can't be digested that's left behind, you open up the bag and it comes back out the same hole. They got one entrance, one exit, it's the same thing. Their larval form is called a planular larvae. These are what's kind of cool is that the planular larvae looks a lot like the placozoan. Just a set of cells in two layers that creeps along the floor creeps along the bottom of the ocean. In some cases, they swim. And so it's almost like you can see that sort of transitional move from what had been the placozoans to what is modern day cnidarians. But Placozoa, so you have to recognize that what we're talking about are all modern organisms. It's not like modern day sponges gave rise to the Placozoans, which gave rise to the cnidarians. Their shared common ancestry is deep in evolutionary time. So the ancestor of all of these things is way, way long ago. It probably looked like a sponge, but it is not a modern day sponge. So you don't want to think about these as like a sponge gave rise to a Placozoan, gave rise to a cnidarian, gave rise to the rest of the organisms. What we're thinking about is the traits that are expressed in these animals now probably represent what was kind of like what their ancestors had. So there's two basic body forms for cnidarians. A polyp and a medusa. Now remember when we talked about radiosymmetrically organisms? What is their orientation? What potential orientations do they have for their way that they're living? Up or down, right? That was it. And we see that up or down view in these two body plans. They're essentially the same thing, a bag with a bunch of tentacles surrounding around the opening to that bag. But they can exist either with the bag facing up, that's the polyp, or upside down with the bag facing down, that's the medusa. And those are only our two forms. But they're basically the same thing, a bag with one opening, surrounded by a set of extensions around that opening. Polyps face up, medusa face down. Okay? If we look across their body wall, regardless of whether it's a polyp or a medusa, we move through, we're going to start inside in the gastrodermis, right? So this is the interior space. It's called the gastrovascular cavity. Gastro meaning digestion, vascular meaning gas exchange. So all of the cells inside the gastrovascular cavity are getting their nutrition from what gets digested in that cavity, and they're getting their gas exchange from the fluid in that cavity, and that's it. Gastrodermis, mesoglia, epidermis. So you have a layer of cells running down the inside on the gastrodermis, and a layer of cells running on the outside, the epidermis, and a non-living acellular matrix running down the middle called the mesoglia. Interiorly, just running below that is a series of cells that form the nerve net. So mouth surrounded by tentacles opens to the inside of the cavity. This is a hydra. Hydra is a polyp-formed organism, so mouth up, attached to the substrate sessile. It can reproduce both sexually and asexually. Here's an asexual bud of a little baby hydra. Imagine that. You just start growing a little baby out of your side, and then the thing pops off and it's free living. That's what they do. They also can form sperm and egg, and those form on the inside of the gastrodermis. A set of cells just all of a sudden start making sperm and egg. And so when they release sperm and egg, where does it get released first? Into their stomach and out their mouth. Their mouth does it all, right? There's where they eat, it's where they poop, it's where they ejaculate. I mean, it's like everything happens out of that one opening. Now, what makes a cnidarian a cnidarian is a specialized kind of cell called a cnidocyte. And a cnidocyte looks something like this. A cnidocyte is basically a venomous cell that has a hypodermic needle attached to it that is going to eject the poison that's inside of that venomous cell into whatever touches it. When you get stung by a jellyfish, this is what happens. It is basically a high-pressure, powered hypodermic needle that jams into the skin of its prey and pumps it full of neurotoxin. Awesome, right? And then once it's attached to its tentacles or hits the thing, it then carries the tentacles and puts it into the gastrodermis where it's going to be digested. So the nematocyst has to discharge to eject hypodermic needle, but it does it in the most cool way possible. This thing has a driver to it that is something you guys already know something about. So I'm going to explain a little bit what happens here. First of all, you'll see that this hypodermic needle-y thing is actually everted. When it comes out, it comes inside out. It doesn't just poke its way out. It everts. It's like when you pull a shirt sleeve through from being inside out to right side out. It sort of slides through to form the thing. That's how it pulls through. So here's what happens. Inside of this cnidocyte is a nematocyst, a specialized kind of cnidocyte. The nematocyst has a whole bunch of venomous, toxinous proteins complexed with ions sitting in there. They're all set up with the ions. When the conditions are right, the conditions are not just having the little operculum get poked. There's often a little cnido sticking up. There has to be a chemical priming that happens first. Okay, so the proteins are all complex with ions, and then when the priming makes it so, the proteins, a protein floods into the space that makes all those ions be released by the proteins that they're associated with. Remember, those proteins are the venomous proteins. So all of a sudden, there's now a whole bunch of ions within the cnidocyte space. Based on osmosis, what's going to happen? Say it loud.
It's going to flood to one side.
Which side? Inside or outside of the cell? Inside or outside of the nematocyst?
Outside. Inside.
High level, remember, fluid follows the solute. All of these ions had been removed from associated with water because they were surrounded by proteins. All of a sudden, boom, they're out surrounded by water. The water forms these hydration shells. There's now low free water on the inside, high free water outside. It all comes flooding in, and that's what pushes the eversion of that hypodermic needle out. And with the eversion comes a expelling of those venomous neurotoxins that had been associated with the ions. And all of this happens in 1/100th of a second. All driven by osmosis. Right? Hit that thing and juice it full of venomous proteins. Pretty tough to be an organism that gets hit by that, especially if you're a little one. If you're a big organism, it just hurts like hell. But if you're a little one, it's not so bad. So, most cnidarians exist in a life cycle that alternates back and forth between a polyp and a medusa, which is kind of cool. For the most part, in most groups, the medusa is the sexual stage. Medusa make gonads, and they spew those gonads into the water column, and mating happens. The polyp is, for the most part, an asexual stage in those that go back and forth. Some groups, polyps do both, asexual and sexual. We'll talk about that in a moment. So, we're going to look at the life cycle in lab a little bit closer, but we're going to look at one right now. This is Obelia. Obelia is in a group that goes back and forth between the medusa stage and the polyp stage. So the free-living, sexually reproducing organism is a medusa. These things are really small, only about an inch across. They swim around, they grow gonads, they spew egg and sperm into the water column. Those egg and sperm fertilize in the water column, then fall down to the bottom after being a free-living planular larvae, forms a colony. Those colonies reproduce asexually, making bunches of buds, or budding bunches of polyps. Each individual polyp is still connected to the previous polyps. Some of them are specialized for making little baby medusa, asexual reproduction. They put out little baby medusa that swim up into the water column, grow into adults, and reproduce sexually. So you find them both as a polyp stage and as a free-living medusa stage. All cnidarians have external digestion. When they engulf them, they're moving them into the inside of that bag. And digestion takes place in the bag, flooding it with digestive juices, breaking it down inside the bag, and then moving the small particulates that are cellular size, the right size to be used by the cells, into the cells themselves. That's phagocytosis, moving the things in. You guys remember that from 200? There's no circulatory system, no respiratory system. No specialized interior excretory system, cells that excrete their nitrogenous waste just directly out to the things, or to the water around them, or into the gastrovascular cavity that's then released. Everything they get is gotten by diffusion and osmosis. There is no like, or adaptive transport across cells. There is no movement from cell to cell to cell. It's each cell gets what it needs from the surrounding environment right by it. There are three major groups. The Anthozoa are polyp only, things like corals and sea anemones. The Scyphozoa are the true jellies. They exist both as a free-living medusa form and a short-lived polyp stage that often does asexual reproduction. We'll look at that in lab. And Hydrozoa, which alternate back and forth between each. They may have a jelly stage that's more common. They may have a colonial stage that's more common. So some of the things that you think of as being jellies are actually colonies. This is the Portuguese man o' war, not an individual organism. It's tens of thousands of individual polyps all grouped together. This float on top is just one polyp. We'll look at that a little closer in lab. The last group of jellies we're going to talk about are the Cubozoa. These used to be grouped with the true jellies, but now we think they're actually their own group. And the reason why I'm going to tell you about them is because they kill lots of people. About 200 people in Australia were killed last year by Cubozoa. Luckily, the time I got stung by a Cubozoan I was in the Caribbean and not in Australia, because it would have been worse. So here's the deal with these guys and why they're so awesome. They school. They swim around in large groups horizontally together. And when you encounter them, you usually encounter a whole bunch of them. So when I got stung, I was taking a night dive, and we started descending down into the night dive, and it felt like somebody jammed a red-hot poker right into the side of my leg. It was one of the most painful things that I've ever experienced in my entire life. I mean, literally, I thought someone had chopped off my leg and then rubbed salt in the wound, right? It just killed. So we come back up, and apparently all of us have descended. There were like 20 16-year-olds, right? We all descended into this thing in one shot. I was wearing a dive suit, and I still got hurt through it. There was one kid who had nothing who got raked across his back, which was just the worst. So what's really amazing about these things is that they have eyes. And those eyes have irises. They have lenses. They have retina. So what does that suggest to you, those of you who know eye anatomy? Things got a lens, a retina, an iris. It can see you! It's a jellyfish that can see you! And it has no brain. It's got a nerve net. Somehow this thing is able to form an image and process it without having any centralized processing place. The reason why it needs to see is so that it can see its neighboring jelly while it schools. Awesome, huh? All right. I can keep talking for